非常开心，今天大家能参加第61期的云端论脑活动。我们首先还是要感谢脑人研各位志愿者的大力支持。我们今天请到了尹路平博士给我们介绍本能行为的神经环路。首先，我介绍一下我们的主持人刘鼎博士，他在中科院神经所获得博士学位，现在在哈佛大学从事博士后研究，关心本能行为以及社会行为的神经环路。我们现在有请刘鼎博士。
好，大家好，欢迎大家来听今天的讲座，非常高兴能够 host 陆平今天的这个 talk。我跟他也是，还有周丽，我们都认识好多年了，有十来年了。然后陆平给我一个基本感受，就是非常刻苦、非常用功、用心，也很拼的一个朋友，一个同学。然后他这成果也是非常显著，他在这边做 postdoc 只有5、6年的时间，就完成了2轮的 postdoc。在非常不同的领域。首先，他是在王帆老师实验室做了一个睡眠跟麻醉相关的课题，然后在林大宇老师实验室完成了今天他要讲的这个关于 female mating drive neuron 的一个课题，都是非常精彩的工作。然后这个他的老板对他有一个评价，我给大家读一下。他说 She managed to complete the entire project from the first experiment to publication in 3 years with the highest data quality any PI can ask for。 我觉得这是个相当高的评价了，而且我刚才看了一下他的这篇 paper 从头到发只用了4个多月的时间，应该说是非常高的 quality。 然后对，就刚才我们说的，陆平现在已经是在西湖大学做 PI 了，所以就是又比我们提前上岸，所以大家可以关注一下他的实验室。我们后面就把时间给陆平。
Okay, thanks for the kind introduction. I feel really happy to see all of you guys here. And I'm looking in a postdoc from Dailin's lab and from New York University. And it's really great honor to be here to present my work published in Neuron, which is about hypothalamic control of female sexual behaviors over the reproductive cycle. So reproduction is widespread in the animal kingdom. Oh, I forgot. So if you have any questions, feel free to interrupt me, and either in English or Chinese is welcome. So the reproduction is widespread in the animal kingdom, and it's the biological process by which each individuals are produced by their parents, and is crucial for all the species. And for our human beings, the reproduction ensures us humans to continue to exist on the earth for all the time to come. However, recently, the Chinese academics deliver a stark warning that China is facing an abrupt decline in population. So there are many contributing factors, but sexual disorder is the most top factor we need to consider about. And compared to male, around 40% of females worldwide have sexual problems. And compared to male, females have more severe sexual disorders. For example, low desire, arousal problem, lack of orgasm, and a painful experience during the sex. And in my project, I hope to use the mouse as a model organism to bring the female reproduction and female sexual behaviors to the forefront of the future lab research. And before jumping into the main study, I would like to briefly introduce the female astrocycles. And there are 4 astrocycles in the female based on the vaginal cytology as shown here. And those 4 stages are proestrus, estrus, metaestrus, and diestrus. And those 4 stages can also be easily visual accessed by the size of the vaginal opening. As you show here, the vaginal opening is largest during the transition between proestrus to estrus, and is narrow for all the remaining stages. And the female will experience ovulation during the pro-estrus to estrus transition period. And during this ovulation stage, female is most sexually receptive. And for all the other stages, instead, female is always trying to dart away, resisting, and then never show any stationary receptive posture. And during this ovulation period, if female is sexually receptive and get ejaculated by the male, she has high chance to be pregnant and later be a lactating mother. So in today's talk, I mainly focus on 3 reproductive stages: diastrus, proestrus to estrus ovulation stage, and the lactation. And lordosis posture is a hallmark of the female sexual receptivity in rodents. The lordosis in Chinese is called 背弓. I will show you this video. You can see here during the mounting, a male mounting, the female back shows this downward dorsifcation, which is indicator of the receptivity. And we will use lordosis quotient to quantify the female receptivity, which is quantified by the number of females showing lordosis divided by the number of male months. And so which brain region are related to female sexual? And for humans, the functional MRI data found that this small area called hypothalamus will light up under different sexual stimuli, includes male pheromones, the viral sexual stimuli, as well as the cervical self-stimulation. And for the recent studies, researchers tend to use rats to pinpoint the exact nuclei within the hypothalamus for sexual behaviors in female. And using the electrical stimulation in the ventral medial hypothalamus, as shown right here, they found that the female will show higher sex score if there is electrical stimulation in this region. And this facilitatory effect is even higher if the stimulation duration extends from 0.5 hours to 5 hours. And based on the literature, females' receptivity are heavily modulated by sex hormones, for example, estrogen. And actually, within the ventral medial hypothalamus, there are tons of estrogen receptor alpha neurons. And people tend to use the dread I to either silent or dread Q to activate those neurons. And they found that when they silence those neurons, the lordosis quotient goes down. However, when they use the drag Q to activate those neurons, there's no change for the female sexual behaviors. Those data collectively indicate that the VMH-BL ESR1 cells are a heterogeneous population. And later, based on our previous lab data, we found we want to screen a more specific molecular marker for within the ESR1 neurons for female sexual behavior. So here we use force staining, which is a neuronal activity marker, and we is shown as green, and we co-stain with ESR1. And we found that actually the sexual related neurons are heavily overlapped with ESR1. And if we take a close look at those false mating neurons, they are heavily confined in the lateral subdivision, but not the medial subdivision. And for today's talk, I will use VMH-BLL and VMH-BLM to represent the lateral and medial subdivision respectively. And our lab also used bulk RNA-seq and found that compared to the VLM region, there are 3 genes CCK-AR, LIGP1 and ES1 are heavily expressed in the lateral subdivision. And we also use the in situ hybridization to verify those data. We found that CCK-AR is the only gene that confinedly expressed in the VLL lateral subdivision. And its expression pattern also quite resemble that of the false mating neurons. So which inspire us to dig into the role of CCK neuron for female sexual behaviors. And in the literature, Xiaohong Xu is a PI from ION, and actually she found that for the whole body CCK-AR knockout females actually indeed show deficits in sexual receptivity. However, those data, those experiments are from the whole body knockout animal, right? So it couldn't pinpoint the behavioral effects to the VMH-VL CCK-AR cells. And indeed, the CCK-AR cells also quite ubiquitously expressed in other social related neurons. And in my project, to establish the specific targeting of the CCK-AR cells, we generate the CCK-AR Cree mouse line using the easy CRISPR. And first, we cross the CCK-AR Cree with S6 to visualize the location of CCK-AR cells. And here, those CCK-AR cells are green, and the ESR1 from immunostaining are shown as red. We can see those cells, the ESR1, the CCK-AR cells are well overlapped with ESR1. And within the lateral subdivision, around 50% of the CCK-AR cells express around 50% of the ESR1 cells express CCK-AR. And all the CCK-AR cells express, almost all the CCK-AR cells express ESR1. And more importantly, we found that those CCK-AR cells are heavily overlapped with the mating force. And both cells confine biasly expressed in the lateral subdivision. And around 60% of the CCK-AR cells express force. And also, for the other way, around 70% false positive neurons express CCK-AR. So based on those data, we are convinced that CCK-AR, at least based on those false staining, it should be essential for female sexual behaviors. And later, we want to propose more detailed scientific questions to answer how those CCK-AR cells are getting involved for female sexual behaviors. The first one is, are those CCK-AR cells essential for female sexual behaviors? So before that, before showing those data, I would like to briefly introduce the three aspects of female sexual behaviors, which I will use it in the later sections. And actually, there are three aspects: attractivity, proceptivity, and receptivity. Attractivity is the female's value for sexual stimulus to induce the male to approach her and close interact with her. For the proceptivity is the female's readiness to approach the male and interact with the male. So it's a different direction. And for the receptivity is the female's readiness to be engaged in the sexual behaviors. So we use the lordosis quotient to quantify to measure female's receptivity. So first, we want to know, are those CCK-AR cells essential for female attractivity, which is the male's present preference toward the female? Here we use DRI or DRI-Q to either silent or activate those neurons with arbitrary answer control. So we use the three-chamber social preference task here, the male is a testing animal, and the two dread eye in expressing female are the testing females, and we randomly select one female as animal of interest, and we will use a preference index, which is measured by the time that the male stay with female one minus the time the male stay with female two divided by the summation of the two. So based on this equation, we can say if the male tend to stay longer with the female one, this number will be a positive value, right? So this is the timeline for this experiment. So we have two females. They will both receive saline injection during the social preference task, and we will get the preference index. And later, for the same day, F1 will receive CLA injection, and F2 will receive saline again, and we will quantify the preference index again. So we will compare the two preference index to know whether the male tend to the male's preference toward the female one before and after CLA injection. Compared to the arbitrary control, we can say that the silencing of those cisplatin neurons has no effect on the male's preference. This was done in estrus stage. We also try to activate those neurons during the estrus, which in the baseline those neurons should be silent. So when we inject CNO, we can activate those neurons. And again, there is no difference for the preference index. So those data collectively indicate that CCKR cells are not essential for female attractivity. And we then want to know, are CCKR cells essential for female proceptivity, which is the female's preference toward the male? And similarly, we use GI, GQ to either silence or activate those neurons. This time, we also use a three-chamber social preference task. But here, we use the dread eye female as a testing animal, and one male and the other female as a stimulus animals. This female is an animal of interest. We will also quantify the preference index, which is the time that this female stay with a male minus the time that this female stay with a female divided by the summation of the two. So we can say if the female tend to stay longer with the male, this preference index should be a positive value. And we will also compare this preference index before and after CNO injection. We first silence those neurons in estrus. Compared to the arbitrary control, we found that after silencing of those neurons, preference index goes down. That indicates that the female tend to stay less time with the male. And we also activate those neurons when the female is in the estrus stage, and the preference index goes up, meaning the female tend to stay longer with the male chamber when we activate those neurons. So those data indicate that CCKR cells can bidirectly modulate female proceptivity. And next, we want to know, are CCKR cells necessary for female sexual receptivity? I will first show you the dread eye. So So based on the CCK-AR whole body knockout data, we can propose that when we silence those neurons, the female sexual receptivity should be suppressed. So here we use estrus receptive female. So female is very receptive before CNO injection. And we will compare female sexual behaviors before and after CNO injection. And here are some video clips. We can see that before CNO injection, this is a male. He tried to initiate a mount and pelvis thrust. So the female is very quiet and stay there and show lordosis posture. However, the same female after CNO injection, she always start away and resisting and never show lordosis posture. This is a summary data. The female's receptivity or lordosis quotient goes down. And more interestingly, we found that we also quantified the approach of the female toward the male. We found that female will approach the male less frequently if we silence those neurons. So indicate that those neurons are not only necessary for receptivity, but also for female sexual motivation. To test the sufficiency of those neurons, this time we inject drug pill to activate those neurons, and we tested in the estrus female. So in the baseline, females are rejecting. So this is the estrus female, so right. So before CNO injection, female is quite resisting and want to dart away, and never show lordosis. And after CNO injection, the same female, we can say that she just drastically changed her behavior, stay there quietly, so male can achieve sexual behaviors. And this is a summary data. So lordosis goes up, meaning that female shows higher receptivity. And also interestingly, we found when we activate those neurons, female tend to approach the male more frequently to allow the sexual behaviors to happen. And besides the estrus, I also tested in the third reproductive stage I mentioned earlier, is in the lactating mother. This time when we activate neurons, there is only a mild increase for the lordosis quotient, which indicate that during lactation, this special stage, there might be more broad circuitry inhibition. So CCK-AR cell activation alone is not enough to increase the sexual behaviors in female. And a quick summary here: when we silence neurons, no change for the attractivity and less for the proceptivity and receptivity. And when we activate those neurons, there is increase in the proceptivity and the receptivity. And next we want to know, those neurons, since for sexual behaviors, it quite heavily rely on the female to distinguish the male sensory cues from the other social stimuli, right? So then we want to know, are CCK cells, the cell respondability differ between different social stimuli? And so to monitor the neuronal activity, we inject ZCAM6S, followed by the implantation of a 100 micron fiber. We test in the pro to estrus female, and to establish a time control manner for the social stimuli, we use this head fixed configuration. Here we have 4 anesthetized social stimuli, one is male, female, juvenile, and toy, and each social stimuli are given 10 seconds for 5 repeats with 1 minute's interval. Here's some demo videos. So we will manually deliver the social stimuli in a face-to-face configuration. You can see here, the CCKR cells preferentially respond to the male sensory cues. We use male preference index to quantify it, which is quantified by the peak signal during the male divided by summation of the signals by male, female, and juvenile. So based on the literature, we also know that during the male-female mutual interaction, the male will elicit ultrasonic vocalization to attract the female and also facilitate the female display the lordosis posture. So next we want to know, are those CCKR cells also responsive to the male ultrasound vocalizations? We also tested in the estrus female and use these half-fixed configurations, and we will replay a 10-second ultra US phase, and we intertwinedly deliver male USV and the combination of the two for 10 repeats. And this is the traces for USV only. We can see that those neurons didn't respond to the USV auditory input. And compared to the male only trials, we can see a mild but very consistent increase if we deliver both male and USVs. This is summary data. So those data indicate that the USVs could enhance the CCKR cell responses to the male olfactory cues. And a quick summary here, CCKR cells preferentially respond to the male cues, and the USVs could promote the male signals. And next, we want to know, are those CCKR cells respond during the female sexual behavior? This is the most, this is the part we are most interested in, right? So here we use the resident intruder behavior task. So we will record 5 minutes baseline and introduce a male into the female's home cage. And after we remove the male, we will record another 5 minutes baseline. So here, during the pro-estrus to estrus female, in those stages, there is successful copulation. So here's some example traces. So we can say the cells shows higher risk activity during the male-female investigation. And during the copulation, let us focus on those behavior video first. So the male first mounted, and then it showed this pelvic thrust. You can see and just one moment, the male just stopped the pelvic thrust, but still clenching on top of the female and slumping to the side, which is the indication of the male ejaculation. And then let's switch to the calcium traces. You can see that those CCKR cells shows highest response surrounding the mounting attempt, but signal gradually decrease during the mount and intermit, and suddenly decrease during the ejaculation, during male ejaculation. And this is a complete trace and the summary plot. So again, we can see that those neurons activate during the investigation and shows highest response during attempt and lower gradually decrease during the other sexual behaviors. And if we take a close look at those behavior annotations, we can see the male, there is always short investigation in preceding the mount attempt. So it's hard. So to distinguish the signals between investigation and mount attempt, to dissociate those signals, we try to categorize the signals. So we categorize into two scenarios. One is a male mount attempt initiated from front. So they have a face-to-face and short interaction before the mount attempts. And the other is the male mount attempt initiated from the back. So there is almost no investigation before the mount attempt. And we can see the constant traces is very similar, but for the red traces, mount attempt from front, they have this trace has an earlier increase, which reflects this. It's induced by the investigation that happened earlier before the mount attempt. Based on the previous video I showed, during the male ejaculation, there seems to be a sudden decrease of the calcium signal, right? So to know whether those sudden decrease is ejaculation specific, here we compare the calcium transients before and after the male introduction and removal with or without male ejaculation. We can see if the male ejaculates, the CCK-AR cell signal almost gone. And however, if the male just achieves intromission and no ejaculation, the CCK-AR cell still exhibits the nice calcium transients. So again, indicate that the CCK-AR cell activity silence is ejaculation specific. So those data indicate that the CCK-AR cell activity is correlated with a female sexual motivation. After a female was ejaculated, she will show less sexual interest toward the male, and consistently the CCK-AR cell signal was suppressed. This is our hypothesis, and to again test this hypothesis, we try to track the CCK-AR cells throughout the whole pregnancy and lactation. So we all know that during pregnancy lactation, female will sustainably show less sexual interest with the male and prepare herself to take care of the young. And here we will each black window is a reporting session. We will report one time before male introduction, one time 5 minutes, 1 day, 10 days, 7 days after male ejaculation, and also 1 day during the lactation. And then we will remove the pups, and the female will quickly resume back to the estrous cycle, and we will report it again. And this is summary traces. So we can see during the whole pregnancy and lactation, CCK-AR cells are also silenced, and the signal will resume back if the female is in estrus again. So again, those data indicate that CCK-AR cells activity coincide with the female sexual motivation. A quick summary here: the CCK-AR cells activate by mutual investigation and mount attempts, and are suppressed by ejaculation. And the next question we want to ask is, does the CCKR cell activity change over the reproductive cycle? So again, we use fibrophotometry to help us examine this question. So each animal was recorded multiple times, at least 1 day during estrus, 1 day during estrus, and 1 day during lactation. And we will use the same paradigm, record 5 minutes baseline and before and after. We will mainly focus on those 4 parameters. One is during the 5 minutes baseline, and the other entry response when we introduce the male into the female home cage, and investigation and copulation. I will show you those data one by one. So during the 5 minutes baseline, before the male introduction, so here is estrus, diastole, lactation. So each red points indicate the peak of each calcium transit. So we can say the CCKR cells shows more calcium transits in estrus compared to the other 2 stages. And then when we introduce a male, so we can see there is entry response, and it's highest in the estrus stage. And if we take a close look at the later traces, we will notice that the CCKR cell also have a sustained elevation of the baseline during estrus. And so the CCKR cells response will be the highest in the estrus, and which because there is a sustained already elevated baseline, so which will make the signal even higher during estrus. And for the last section, we want to examine the electrophysiological mechanisms underlying the in vivo data. So here we use SNAP recording, and we cross the CCKR with AN9 to visualize the CCKR, to help visualize the CCKR cells. So first, in the current climb mode, we record spontaneous activity of neurons. So CCKR cells show most spontaneous firing estrus. And then we rupture the membrane and record it in the current climb mode, and to record the FI curve. And again, those neurons fire at most frequencies in estrus. And we will also record this synaptic inputs. We record, this is spontaneous iPSC. It's the cells receive, we can say the cell receive least iPSC in estrus. We also record the spontaneous EPSCs, and actually those cells receive very rare EPSCs and also didn't show any change across the 3 stages. And a total summary here. When we silence those neurons, receptivity goes down, and when we activate neurons, it goes up. And the CCKR cells could be preferentially activated by the male cues, and the USB could promote it. And for the free moving task, found that CCKR cells could be activated by the investigation, and also the physical touch of the back muscle by the mount attempt, and it could be suppressed by the male ejaculation. And for the SNAP recording, CCKR cells shows higher spontaneous activity during estrus, and more excitability and less EPSAs during estrus. So for the functional implications, we know that in human, especially in female, always show, especially after the monopause, it shows a low sexual desire and painful in sex. We hope our findings could find a way to promote female sexual desire and also relieve the pain during sex. And the last slide, the significance of our study, we update the current review about sexual dimorphism in the ventricle. In the early studies, people know that ESR1 is essential for female sexual behavior. And 2019 data, they found that activate those neurons, female axis no change, indicate that indicate that ESR1 is a heterogeneous population. And with false and other behaviors, we found that the mate-related gray cells maybe reside in the lateral subdivision. And those neurons express CCK-AR molecular marker. And recently, there is another paper they found by David Asson. They found that the female fight-related cells actually reside in the medial subdivision. There are two separate populations. And however, in the male, the fight and the mate cells intermingle each other. There is no boundary between the two. So finally, I want to thank Dari and also other lab members, and also my funding resources, and also my previous lab from Duke University, and also from Dr. Yushen Shu from Institute of Neuroscience, Shanghai, China. So I would like to thank everyone and take any questions. Thank you so much. And any question is welcome.
好，那我们就回到报告。很感谢林路平的非常有意思的一个研究，感觉很多点都可以再深挖下去。那我们现在就进行一些和观众的交流。如果大家有问题的话，就举手，我来叫大家，然后大家开麦来问就好了。刘明哲先来吧。还在吗？
我可以听见吗？
可以。
我这边摄像头为什么坏了？不管摄像头。
没事，你先问。
Okay, thank you. A very nice talk, and I learned a lot from the female sexual behaviors. So I have several questions about the input and output of these neurons. So actually, these neurons, CCK receptor positive neurons, they are in hypothalamus, VMH. First, I wonder how they can sense the information from male. Which one do you think? Which type of sense do you think are most important? As we know, the female can feel the male, mount them, and they can see them, can hear them, have some vocalization, or they can smell the male sense. So which one do you think are most important? Do you know which region send projection to this region?
Only one question. Okay, so thanks for the question. So first of all, input, we use retrograde reverse virus treating and found actually cis-parent cells mainly receive input from the local hypothalamus. And since those neurons express CCK-A receptors, so for the input, we mainly focus on the cells that express CCK, and we found that there are some interesting regions. But this is my current work, so I think I will keep secret. But and yeah, and for the output, we also use GLP treating, and I show those data in my supplementary figure. It mainly project for the output, it can project to very diverse regions compared to the input. In the future, it might be interesting to study the output in the MPOA and PAG and MEA, since those regions have been reported to be essential for female sexual behaviors, especially the PAG is very important for the lordosis posture itself.
Got it. And which type of function do you think these neurons perform their functions? Do you know? Do you think it's about reward, decision making, or just motor functions? If you activate these neurons, they can facilitate sexual behaviors. So, which type of function do you think these neurons have?
This is also. So first, for the rewarding, we use real-time place preference task, and we found that when we couple the blue light with to activate those neurons, it's very rewarding.
Very rewarding. Okay.
Yeah, it can also promote the movement. The behavior speed. Yeah.
Maybe both the reward and motor are important for the sexual behavior.
However, when the female singly in her home cage, when we activate those neurons, the female never show lordosis posture. So that indicates that those neurons are not didn't encode the posture, lordosis posture itself. Here we have a review paper. We have some speculations. Here, we think the CCK's self is the subcutaneous is instantaneous activity didn't encode the lordosis, but it can summarize all the inputs from other regions. For example, the MEA, BNST can encode the male sensory cues, and so when the cis-pair neurons submit those input from other regions, it can decrease the threshold for PAG neurons.
So these neurons are GABAergic neurons.
Vimageville are augmented neurons. In summary, the cis-pair neuron itself didn't encode lordosis, but it can modulate the threshold for the PAG neurons. And the PAG neurons could control the lordosis directly.
Okay. That's great. Yeah. And another thing is very interesting is that when the female feels ejaculation or another terms, so these neurons are inhibited. So I think maybe some other sensation can send an input to region, this type of neuron.
Yeah, and we use you asked the input, right? And we found the CCK positive neuron is also GABAergic, so which might play a very important role to control the sudden decrease during and after ejaculation.
Do you know what's the function of this receptor? CCK can activate the neurons or inhibit the neuron?
Oh, sorry, can you say it again?
CCK, what's the function of this type, the receptor, CCK-A receptor? CCK can activate the neuron or suppress the neuron.
Oh, so we use some pharmacological cannula injection. We inject either CCK-A receptor agonist or antagonist, so it can by directly modulate sexual behavior. So yeah, that at least indicate that CCK are signaling. This papanergic signaling is very important for sexual behaviors. And here, with this figure, I want to highlight the importance of the papanergic neuron signaling. Here we can say when we evoke electrical stimulation, actually there is a delay around 20-30 minutes. And when we eliminate, like remove the electrical stimulation, the facilitatory is long lasting actually. So that indicates lordosis posture itself is controlled by some hormone or peptinergic.
OK，
that's
it， 多谢师姐。
Thank you so much.
OK，so 我们后面是卓清、谭涛，然后是李明娟，请大家把时间控制的稍微短一点。好，卓清。卓清还在吗？那我先插一个问题啊，看看后面，就是在 parenting 的时候，或者他那个 pregnancy 的时候，这个 neuron 就 activity 非常低，它是因为 input 变少了呢，还是它的本身的 excitability 变低了呢？
我们最后的 slide recording 应该是都有，就 intrinsic 和 extrinsic synaptic 都有。我们可以看到 lactation 的时候，这个 neuron 本身的 firing 是降低的。我没有试过加那个 synaptic blocker， 但是我觉得应该加上 blocker 也是一样的结果。然后其次就是它的 EPSA 很少，而且没有变化。然后 iPSA 的话，在 lactation 阶段有一个极大的上升，所以我觉得我不知道它的 input 的就是脑区数或者是 input 的 cell number 是不是有变化？但是这个是可以今后去做的。
所以它是一个 disinhibition， 然后让它导致的兴奋，它通常状态下是一个被 inhibit 的状态，是吗？
通常状态是
就是你的 iPSC 会变少嘛，在 estrus 状态下，所以它在不是 estrus 的时候就是被抑制的，是这个意思吗？
是。
OK， 好，我们后面接着问吧，就按我刚才说的这个顺序。
我现在能听到是吧？
尹
博士你好，就是我想问一下，因为我看做了很多 estrus、diastrus， 好像 med-estrus 也有涉及，就是我自己是在做 pre-estrus 相关时期的一些表征嘛，印象中 pre-estrus 那些神经元的活性都特别高，就是想问一下你有没有设计去测一下 pre-estrus 时期的这些东西？
我在我这里面其实我没有分 pro-estrus 和 estrus， 我就是通过它的 smear 看到它只要是 pro-estrus 或者 estrus 我都记，然后如果是female 它可以它有很大的概率是 receptive， 但是也有比如说2-3成的概率是不 receptive 的，反正我就用 receptive 的 data 来分析。
不服气。好的，谢谢。
那该我了是吧？
你好。
好，那个尹博士你好，非常漂亮的工作啊！我接着刚刚那个问题问啊，就是说在这个期间呢，它这个本身的激素，一个是雌激素啊，肯定是有很大的变化的。然后呢，另外呢，在那个孕期呢，又有孕激素有个大量的变化。有没有探究这个激素啊，在这期间对它的一个大量的影响啊？或者是直接就通过这个激素注射激素或者影响它的激素的表达，看一看这部分脑区它的神经兴奋性的改变，这部分工作有没有开展，或者接下来会不会开展呢？
谢谢你的问题。首先就前人的工作有一些提示，就是说当把卵巢切除就 OVX 以后CCKR 表达是几乎为0的。然后在我的这个课题里面我没有受这份 data， 就是我发现 OVX 以后我用 GQ 把这群 neuron 激活， female sexual behavior 依然是增加的。这就意味着就正常的时候 natural 的状态下，就是这个雌激素它会作用于表达雌激素受体的 CCKR neuron， 让它激活，进而促进这个 sexual behavior。但是即便 就是没有 estrogen receptor， 没有 estrogen，通过就是卵巢切除没有 estrogen， 但是我们人为的去激活这群 neuron， 它可以 bypass estrogen， 直接投射到下游来发挥就是promote sexual behavior 的作用。就是说 natural 的条件下 estrogen 还是非常重要的。如果没有 estrogen，
那个后面哺乳期间会不会就是它这个降低啊？这些是不是因为它的孕激素这部分增加了导致的，对吧？这部分有没有相关的解释呢？
我们有一些 sequencing data 有提示 lactation 阶段 CCR 的表达量是下降的。
好，第二个问题啊，就是说在这个交配期间呢，就是实际上它是有这个昼夜的影响的，包括它的 arousal。就我想请教一下，就你在做这个行为的期间呢，有没有发现，比方说白天、中午和傍晚啊，这个对它的这个行为有大量的这个影响？
我其实没有发现，但是 Harvard 有一个实验室，他们发现是跟这个昼夜circadian 有关系。他们发现就是下午1点钟左右，那个 sexual lordosis 是最明显的。我目前没发现。
就是你现在做这个行为是这个就是没有选定特定的时间做这个相应的行为。
我是在白天做，但是我们那个鼠房是 reversed light。所以鼠房的老鼠是晚上，就是我们的白天它是晚上，然后我们做实验的时候都是把灯关了，用那个红外的 camera 来监测小鼠的行为。
好，
我是偏random 做，就哪有时间就在哪个时间段，我们不觉得有区别，明显区别。
OK， 因为很 strong 啊。last question 就是在这个。你发现了一个非常interesting 现象，就是 directly after ejaculation， 对吧？然后一直持续到这个 lactation 都没有恢复，然后你没有继续往后面做呢？什么时候这个又恢复了呢？是不是跟他这个在后面 lactation 结束之后，或者甚至是在 lactation 结束之前，它就发生这个recovery 这部分有没有做呢？然后呢，在这期间呢，实际上我们以前也很多文献报道，就是说在lactation 这个期间，它是非常 aggressive 的 female 只有在 lactation 期间它是非常 aggressive 的。你当然没有在这期间就是同样这个脑区没有，实际上也有很多文献报道，包括你们以前实验室也是报道，就是这个跟 aggression 非常相关的。会不会这部分神经元在这期间从它的这个 sexual arousal 变成了这么一个 aggression， 或者它有一个目的任务态的一个转变？不知道你对这方面的一个理解是什么样的？
切换到这张 slide。就是根据 David Allison paper， 就是 female 在 lactating 阶段 aggression 的时候，激活的 neuron 是另外一群，就是位于 radial subarachnoid， 它表达是 NPY2R， 然后他们用那个 NPY2R negative ESL1 positive， 把这群 neuron 把那个 NPY2R negative ESL1 positive neuron 激活以后，也可以促进sexual behavior。然后同样呢，我发现我这群 neuron 如果激活以后，不仅可以促进 sexual behavior， 同时可以很有效的抑制aggression。然后现在有趣的是，这两群它都是 glutamatergic， 然后我们叫这个圆圈我们叫它那个 core， 然后旁边还有一圈 shell， 这个 shell 是 GABAergic neuron。然后这两群 neuron 它在这个 virgin 和 lactation 是这个行为是如何转变的？是通过 long range 的 GABAergic， 还是周围的这种就近的 GABAergic neuron？这是今后我们想研究问题。
好，谢谢啊。
尹老师您好，可以听到吗？
可以。
非常精彩的工作，就是我想请问一下，您有做过光遗传激活VMATRL 的 CKR 神经元吗？因为以及包括 不同频率的光刺激激活。就是在这张 PPT 上，您也提到，这个 VMHVL 它是一个抑制性的核团。我记得，大卫的安德森的实验室，他们应该有发表过，就是低频率光刺激 VMHVL 它可以诱导出 male 的 mount 或者是 mating 行为，但是高频刺激的时候诱导的就是 aggression 行为。对，所以想请问您一下。
我在 paper 里面有用 optogenetics 来重复这些实验，然后我就发现 optogenetics 我通常给20秒，大概是4毫瓦到5毫瓦的光强，然后我发现就是male 的mount 和 intermission 的成功率会增加，但是 female 依然是想要跑，但是 somehow 他没跑走，没有可以追上他，就是 female 依然是 resistant 的，他没有受到 loss exposure，就是说 optogenetics 的结果就是没有 drag kill 那么显著。我这里面给出的解释是因为就是说刚才我有一个 hypothesis， 就是 VMHVL ESR1 cell 它要把它的这个它的兴奋性一旦增强以后，它需要输出给 PAG， 让 PAG 做一个最终的 summation 来 induce the loss exposure， 这个需要时间。所以 drag kill 我们激活了30分钟，就是它有足够的时间让 PAG 去激活，但是 optogenetics 它时间还比较短，所以它的效果是不显著的。就是没有依然会有更多的 mount 和 sexual， 但是 female 还是想要跑走，所以它没有那么强的 lordosis 的表现。然后还有那个不同的光强，这个我没有试过。
好的，谢谢。
好，文杰。文杰在吗？
能听到吗？你好。这个我其实有一个很 general 的 question， 然后按照知乎的优良传统，先问是不是，再问为什么。所以就是那个是不是就是说 ，is it true that in general females find sexual activity less rewarding than males？ 然后为什么就是what do you think was the biological mechanism for that？ 是不是因为这个 rewarding 的这个 processing 的过程，或者说它的 circuit 在这个 female 和 male 之间是不一样的？
我觉得肯定是有区别的，但是怎么样去 quantify 他们的这个 rewarding 的大小，我不知道。就是、肯定是有对，就是对于 female 来说 sexual 肯定是 rewarding 的，所以说在 estrus 阶段的时候 female 会更多的去 approach male， 想要让这个 sexual behavior 去发生。但是相反在那个打 estrus 的时候 female 很少主动去接近 male。同样呢，我们用那个 three chamber social preference task， 我们也发现 estrus 的时候female 会花更多的时间跟 male 那个 chamber 待在一起，但是打 estrus female 它会花更多时间留在那个 female cage， 所以肯定是有区别的，但是我不知道怎么去 quantify male 和 female 的这个差别。
我记得你们实验室最近那个 dopamine sensor 那个 paper 好像 male 的 response 要高一些是吧？ female 会低一些在 mating 的时候。
这就是我刚刚想说的，被就有点截胡了。就是说，是不是可以直接去检测这个脑内这些rewarding 的 signal， 比如说 dopamine 嘛，当然说你也可以说这个其实不是一个 rewarding signal， 但是大家就是认为的 rewarding 的或者说就是愉悦的这些 signal， 如果去检测的话，会不会就是说可以用这个作为一个指标来看？
我觉得不同个体还是很难的吧，比如说 female 的 dopamine neuron 可能本来就少一些，所以它信号就弱一些。那我不确定你通过这个信号的大小直接比有可比性。
你可以做一个 transition 的 assay， 在中间做一个 pair 的那个第三种 reward， 然后你让 male 去选 female 和那一种，然后去 female 去选male 和那一种，然后做一下换算可以，就是那个 banana juice 跟 orange juice 那个叫 monkey 的那个 study。
他们有发现，就是在 female 前面放一张那个电网，然后电网的对面是一只 male， 然后 asterisk 的 female 它会更倾向于 take 这个 risk， 经过那个电网去接触对面的 male。对，还有一个，对，我们 lab 那个 dopamine 那篇 paper 好像有发现，就是说 asterisk 阶段其实 female 大部分时候是receptive 的，但是也有可能性是不 receptive， 尤其是 asterisk 的那个后叫什么 late phase， 它是不 receptive 的。如果是 late asterisk female 不 receptive 的话，它呢也没有 dopamine， 它的dopamine 信号也很弱。所以就是说，即便都是 asterisk， 它的dopamine 可以很明显的指针这个 female 是不是享受这个sexual behavior。
OK， 谢谢。
对，我们还有一个非常有趣的设计，就是我们设计了一个那种类似于 open arena， 然后在open arena 的中间放一块隔板，隔板下面掏一个小洞，那个小洞因为male通常 size 比较大，male穿不过，但是 female size 小，只有 female 可以穿过。然后我们就会看，就是激活或者抑制这个 female 它本身穿到 male 的那个 chamber 里面的可能性有多大。所以这样的一个设计的话，它可以把那个 male 的成分，male 起始 sexual behavior 的这个成分给去掉，仅仅体现 female 的参与。我觉得是更直接的一个证据。今后我们就倾向于用这样的一个 design 去做实验。
OK， 大家还有别的问题吗？我再问一个问题啊，就是你一开始提到的那个，就是性功能障碍，我觉得这个非常有意义的方向。现在在动物模型上面，或者说有没有什么研究思路啊？还是这是你的秘密不能说？
不会，没有，就是有很多就是 gene mutation 的 model， 但是大部分都是作用于那个 kiss-peptin 1 neuron 或者是 GnRH neuron。然后
或者我换一个问法，在人上面的这些问题，它到底是中枢的呢，还是外周的？它是哪里出问题了？知道吗？
都有啊。
都有。所以现在的这种疗法，这种 drug， 它的 target 是哪里呢？还是没有 drug？
其实很多就是增加 sexual motivation 的药，有 target 五羟色胺的，NE 的dopamine 的，但是都有副反应，所以
就是它是一个比较广谱的，但是不是特别 specific。
我目前我觉得那些药其实都不是特别 specific。
那你这个 neuron 有可能会设计成一个靶点吗？或者说你去做一个 CCK 类似物呢？有这种可能性吗？
如果可以CCK 在全脑其实个数都有表达。如果可以涉及到一个就是只能 target 到这群 neuron，我觉得还是很有意义的。然后我们现在也有一些，比如说把 CCK2 敲掉，然后看它对 sexual behavior 或者是那种 HPG 轴或者是激素的分泌，我们发现其实都是有影响的。就是它不光能够调节中枢，它也可以调节外周的那个整个 hormone regulation。它是就是叫一个 central hub。
OK。好，大家还有什么问题吗？我再问个问题， 你有没有试过就是 Liren 叫这群 neuron？ 我看其实就非常小的一群，大概你估计有几百个吧就。
我觉得 Liren 太 dirty 了吧。
好吧。
但是有 Liren VMH 的，那个肯定是receptivity 下降的。对，我有一个技术上的细节，然后现在可以讨论一下，就是我一开始是埋那个400微米的 regular fiber 做 fiber photometry， 然后我就发现这些 neuron 因为它在最底层嘛，就经常会死掉。我们试了很久，最后用那个100微米的 fiber， 然后细胞就可以很好的活着。但是就是旁边好像 NPY2R 和 ES 旁边的 ES1 好像那群 neuron 就比较 resilient， 不会死。反正 CCK neuron 就还蛮神奇的，特别对这些机械刺激，蛮 sensitive。所以整个的一个 summary 就是以后大家如果做这种特别深层的细胞容易死的话，就换成比较细的 fiber 就可以。
OK， 我还以为你说你把它搞死了以后，它的 behavior 会有变化。Anyway，
我没有做过 in situ， 就是没有好的 CCK antibody， 我没有做过 in situ， 我不知道它到底还活的。我有时候想的是，我 virus 本身也没标记全，但是我有用那个 DTR diphtheria toxin 去尝试杀死这些细胞，就是也有一些比较有趣的结果，那个就不太方便在这讲。
那你是怎么判断就是那些细胞它死了然后用400微米的时候？
就是我不埋 fiber， 它就活得很好，我埋了它就双侧对照嘛，一侧埋另一侧不埋。
就是你是双侧都打了那个 Gcamp 的病毒，然后就是你埋了之后它就没了。那好奇怪呀。我们实验室都用400的，然后反正都能记到信号。
所以我就说，哎，你们是记得 CCKAR 吗？
对呀。
这个我也不知道，因为我来这个实验室加入实验室之前，其实也有其他的学生或者博后在做这个课题，但是他们都放弃了，就是因为这个 fiber photometry 记不出来信号。我也尝试过一段时间用400微米，就是也不行，最后才用了100。就是我是说，这不光是我个人的问题，就是我们整个 lab 的其他几个人也有这个问题。
我们实验室有好几个都是用了400，然后都可以记到。
我不知道，没关系。等我回国了，我准备买你们 lab 的 CCKAR 了，到时候就可以试一下，是不是你们的那个 query 做的好。但是我发现我们反正都是用那个 easy CRISPR 方法是一样的。
好的，如果没有问题的话，周丽。
好，我们感谢我们今天主持人以及嘉宾李卫平博士精彩的报告，感谢大家。然后接下来我会结束录屏，大家可以再闲聊几分钟。等一下啊 ，stop recording。